Okay, so we are on slide number 29, and we can now say that the 5' and 3' untranslated regions are related to longer exons than those that code for protein. Um, so we see that those regions are actually bigger than the actual regions that code for protein. And there might be a reason for that. I mean, those are the starting and ending ends of a gene, and we want to make sure that we can recognize that. So always remember that there are other factors in a gene besides the protein coding sequences. So when we look at a gene and we compare the length of the gene to the length of the mRNA, usually the gene is about 5 times longer than the mRNA, okay? Because we're taking out sequences that are not needed and will interfere with the translation process. And when the ribosome gets the mRNA, we want it to be specific and extremely collinear with the peptide or the protein that has to be produced. Okay, so that's really, really important. So just imagine how much taking out of sequences and splicing and cleaving is going on before we have that mRNA. Alright, I'm going to skip on slide 30. We just said that. So we're on slide 31. Although usually we don't see this happen, there are examples of one gene coding for more than one protein or function. Okay, such genes are called overlapping genes. So if the gene codes for a complex that we've talked about made of subunits, or if the full-length protein has also a shorter version of itself, we might find different genes that code for it. Okay, we are on page 32, and now on slide 32, 33, and 34, we're going to go over a few examples of different situations where genes can overlap. So we see in example 1, we see an example of full gene overlapping. So for instance, we have the upper strand that is going from the left to the right, from the 5' to the 3', right? And we have the lower strand that is going from left to right, from the 3' to the 5'. So they are what we call anti-parallel, right? And we already used this term. So the upper gene, you can see, is way longer than the lower gene. So when you look at the lower gene, we have full overlapping of the upper one over the lower one, right? In slide number 33, we have example 2, which is called tail to tail, right? Because overlap. So we see that the overlap is in the 3' ends of both genes. Okay? And naturally, the third example is head-to-head, meaning an overlap of the 5' ends and the 5' ends. So those are the three options that we have. Such cases can be found in the human body regarding mitochondrial DNA, abbreviated as mtDNA, and we will talk about mitochondrial DNA. So the overlapping tends to be short, okay? It's not long periods or series of overlapping. And it usually retains to a very unique coding function. So it doesn't happen with all genes, only with those that are related to some kind of unique function that is needed. Some genes allow what we call alternate gene expression patterns by affecting the exons. What does that mean? That means that we can have a change of exon content at the level of mRNA. Optional only exons, meaning that they don't necessarily have to be expressed. And mutually exclusive, meaning that it's one or the other. So think about switches, okay? You go into a room and you have a panel of switches and you have different lights. How many different combinations of light can you produce? So you can work each switch individually, right? Some switches may not operate when the other ones are operating, right? And some must work when others work. So you have all kinds of combinations, and those are the three options that we're talking about here. So basically we can switch on in some ways exons, and some are limited in how they can be expressed, and some must be expressed at the same time. It all depends on the function that you need in the cell. So such alternative proteins are going to have parts that are common to each other and parts that are different. When we produce changes to the 5' and 3' ends, we will produce the same protein, okay? But will affect the regulatory aspects. So since we're talking about changes to the ends, we're affecting the regulation of expression and not the protein itself, because if you remember, the protein coding sequences are where? In the middle, okay? So it's just common logic. Even if axons are switched, meaning that they're going to move places, it's not going to affect their function, because the function depends on the amino acids, right? And the amino acids depends on the sequence. We didn't change the sequence of the axon itself. We're just moving it from place to place, okay? So assuming that sequences have moved, but the reading frame did not change, we shouldn't have a problem. Does that make sense? I hope so. So when a protein has a few patterns of expression, for instance, alpha, beta, and so on, and there's a lot of examples in the body. If I'm not mistaken, the book gives you at least 2 examples, so notice that in the book. One can be expressed more than the other, or even only instead of the other. So proteins can have certain variations, or they're called patterns of expression, right? And sometimes we see all patterns expressed in different amounts or ratios, and sometimes one is expressed rather than the other. We're on slide 37, okay? So this is due to a process called alternative splicing. So remember, we can splice a gene. We can take out parts, we can put in this case splicing is taking out, we can rebuild, recombinate, right? So this gives us a lot of variation between the outcomes that we can produce, right? So for instance, you have a very good example in figure 2.20 on page 44 of the book, and I advise you to read it. If an axon is not recognized for some kind of reason, it might be spliced out, right? And that will produce a different product it might be regarded as a pattern of expression. Okay, on slide 38, you are referred to a movie. Enjoy, have a bit of popcorn or whatever. So, even though two proteins can be the same at their ends, one might be perfect and one might be lacking an axon. Meaning, when I'm saying protein, that correlates to the genes, right? So there are 60% more proteins than actual genes. I'm going to say that again. There are more proteins than genes. 60% more. What does that mean? It means that alternative splicing is the way to create greater variation in proteins than genes. It's actually very efficient. Alright? Of course, if it was not that way and we were talking about one gene for one protein and we did not have those mechanisms in between creating variation, our genome would be even greater, even bigger, right? Why is this important? Well, first of all, it's very sophisticated. 2, it's very efficient. And 3, remember, the cell does not have unlimited space. So by using alternative splicing, we are saving space for the cells. Is that important? I believe it is. And we'll actually touch that point and talk about it in future presentations. Of course, when we're talking about alternative modes and splicing, or alternative modes of gene expression, which is the same name for what we were just talking about, this will likely change the sequence, right? Makes sense. Okay, we are on slide 40. So, one of the major questions asked in the field that you see on slide 40 is, can a single axon be responsible and identified with a certain function? So let's discuss that a bit. In some cases it's possible, especially in the immune system, for instance. The immune system is very specific. It has specific factors and features for specific defense mechanisms. So that is relatively easy. Or let's phrase it this way: if there's ever going to be an easy case identifying and connecting, a single axon with a specific function, it will probably be in the immune system. So yes, it's possible. Alright, so we are on slide 41. So for instance, we have immunoglobulins that are made of combination of different chains. Alright? And you can play with them. It's like playing with Lego. Biological Lego. It takes several genes to code for the whole complex, meaning that every gene is contributing a different subunit or component of the whole complex. So certain axons have been associated with certain functions, such as the secretion of insulin, for instance. Okay, so they are an amazing family, allowing us to actually connect between a gene, or an exon, and function. When a cell receptor has various functions, it will be apparent in the exons, in the gene. So just remember that, the cell, the membrane of the cell and other parts of the cell has receptors, and sometimes one receptor is able to conduct different or fulfill different roles, right? So that has to be presented, be represented in the exons, meaning in the gene. So different functions are often regarded to as modules. So we'll say that a protein has different modules. A receptor has different modules. The gene, the exon, has different modules. Okay, so you might encounter that term too, and it's explained in the book as well. It is possible with time for the modules to have evolved to create different functions. Okay, so not necessarily all modules existed at the same time, and some are part of evolution. We can actually identify recent modules that were added to genes, okay, through evolution. And when we say recent, do you remember what we talked about evolution? We're playing a game of millions of years, right? So recently is not in your lifetime, not even in mine, not even in our lifetime combined. Okay, one way to identify those recent added modules is because usually when a component has been added recently in evolution, we'll find it on the surface of the protein. So theoretically, the older the component, the more inner it will be. Okay. Okay, so on slide 43, we are saying the following: the more modules in a gene, the more functions a protein will have. Okay, so we are associating modules with functions. All right, we'll stop here and continue in our next video. That will also be the last one for this chapter. Hang tight.